So, Mary Beth has talked to you about some of the health effects of climate change on kind of a more global situation. We're going to talk a little bit about some of the studies that we're doing on a more local level that tie into this. And, touchy. Okay, so we've talked about, some of the problems with water quality and climate change, but also a lot of the benefits that you get from living near the coast. For all the concerns we have about the coast, there really are a lot of health benefits from the oceans and from living near the coast, and we want to try to preserve those as much as possible and maximize those for the populations living on the coast. One of the challenges we have in microbial water quality is the discharge and the fate and persistence of various microbial toxicants that come from wastewater discharges, from coastal inlets, from septic fields, cesspits, a variety of sanitary infrastructure and the man-made built environment that gets exported out into the coastal zone and can have an impact on ecosystem health, on the health of reefs, fisheries, shellfish, as we were just talking about, and of course human health. Now, throughout the country, especially in recreational waters, we have a National Beaches Act, and most county health departments are mandated to assess at least some of the recreational waters and shellfishing waters for particular microbial contaminants or indicators of microbial contaminants that could impact public health. The challenge, we're going to talk about the challenges of those traditional methods, but they need some help. So one of the things our lab is involved with is the development of new methods, genetic molecular methods, to enhance the current techniques that are used to detect and basically tell where a lot of these toxicants are coming from. This is important, especially in the face of climate change, because we have issues right now with the discharge of these microbial contaminants. But if you think about the inundation maps Dr. Enfield talked about, as sea level rises and we get more extreme events, more recent flooding, more frequent flooding, changes in tides, all of these things are going to impact sanitation, our septic systems, landfills. As these areas get flooded, there's going to be lots of new sources for the discharge of microbial contaminants, and it's going to change the patterns of transport and the fate of a lot of these microbial contaminants. So we're working on developing methods and applying those methods to measure how these microbial contaminants are changing, how they're being transported, and their persistence in the environment. And it turns out that South Florida and the Florida Keys is an ideal area to test these methods out because of the variety of sanitary infrastructure you have, your elevation. You're going to be one of the first areas seeing a lot of the impacts of sea level rise and climate change. So it's a good place for us to work on these methods. So in the traditional sense, looking at microbial water quality, they use fecal indicator bacteria to judge when and where microbial contamination and disease-causing organisms are occurring in the coastal water. A fecal indicator is simply an organism that's commonly found in the gut of people and that's shed in your feces. Now, an indicator organism in and of itself doesn't necessarily cause disease. So everybody has fecal indicators in their gut, everybody sheds them, and if you find them in the environment, the presumption is that's caused by some kind of fecal contamination. The reason we test for fecal indicators is that if somebody happens to be ill in the population, they will also shed pathogens along with those fecal indicators. If there's another person who has a different kind of disease, they will shed those with your fecal indicators. And it can be very hard to test for specific types of pathogens. They may only be in a very small subpopulation of people. They may be very hard to test for or very expensive to test for. And they may be infectious in very small doses. And so since we can't test for every possible disease-causing organism, we make the assumption that if you see especially human fecal contamination, there's a good chance you could find disease-causing organisms from sick individuals. So our best way to keep the environment and the population safe is to prevent human fecal contamination in the environment in the first place. So we try to avoid this mix of pathogenic microorganisms in sewage and try to prevent sewage and septic contamination to the environment. Now, the ideal indicator should be one that you can find in human feces, that is only from human sewage or septic sources. It typically doesn't cause disease itself, but it reflects and mimics the environmental fate and persistence and transport of the pathogens. So it's a good proxy for the pathogens. And there are several different kinds of fecal bacteria that we use as indicators. Right now, the two groups that are used for regulatory purposes are coliforms, the total coliform group, and enterococci. So in the state of Florida, every state develops its own criteria based on EPA recommendations. In the state of Florida, we use enterococci and fecal coliforms. And in marine waters, we primarily use enterococci. In that case, we don't want to find more than 103 of these cells of enterococci in any particular 100 mils single sample. And if you go and you sample 5 times and take an average, you don't want to find any more than about 35 of them. Now, this current methodology is based on growing these organisms up in culture, in media, on agar plates or in broth. So the two primary methods are filter plates. This is based on passing a water sample through a filter, placing it on an agar plate that grows the bacteria up and turns it a particular kind of color. If enterococci are present, they turn this nice blue color, and you can count them and determine how many there are per a given volume of water. The other regulatory method is the chromogenic substrate method put out by a company called IDEX. It's a most probable number test where you mix your sample up with a broth and you put it in these little trays, and it just replicates a whole bunch of them, and you look at, is there growth and does it turn fluorescence, and you count them up and it'll tell you how many of these bacteria you have per 100 mils. So both of these are specific to enterococci. The challenge of these methods are that, first of all, they're culture-based. That means you've got to be able to grow the bacteria up. And that typically takes about 24 hours. So it doesn't really tell you what the water quality is right now. It tells you what the water quality was yesterday. Which, if you're working on a beach or some other area or shellfish bed and you want to know what the situation is right now, you don't know that. The other problem is it has no source tracking capability. So it turns out there may be other types of organisms that shed these, or they can be found in the sediment, or they can be found on biofilms of plants and things. So they turn out not to be a great indicator in warm subtropical waters, that they violate some of the axioms of an indicator. So, some of the problems, it's unclear if you're really testing recent fecal contamination. They're too slow, they don't directly test for pathogens, and they don't have an ability to source track, so they can't really guide your remediation or fixing of the events. So what we do is we add another method to enhance that. We use genetics to look for the DNA of specific strains of fecal indicators that you can track to a certain host. So we can look for different alternative indicators, such as the Bacteroides group or others, that we can say this fecal indicator specifically comes from humans, or horses, or cows, or pigs, or gulls, because that host has evolved with that particular organism, and we can genetically type it to where it comes from. This allows us to discriminate sewage and septic from terrestrial runoff, from agricultural runoff, and other such sources. So this allows us to do species-specific identification, to do it very rapidly, sensitively. You can test for multiple targets at the same time, and you can source track that back to the host. So these are some of the different alternative indicator organisms we use. We'll talk mostly about Bacteroides today. That's the primary organism that is being used right now to type to different hosts, and that's one of the alternative indicators that's moving rapidly towards new regulation for testing water quality in recreational waters and shell fishing, recreational beaches, and so forth. So in a comparison briefly of the three methods, you have the plate counts, where you count blue colonies, but you got to grow them up over 24 hours. You have chromogenic substrate, which is the most probable number test, also requires growth. And you have molecular methods. The one that's the front runner right now is something called quantitative polymerase chain reaction, or PCR. You might have heard about PCR. It's used a lot in clinical situations. So there's all kinds of medical tests that use this technology, and it's now coming into environmental testing. We do this so that we can develop new products that more rapidly and reliably biosense and can provide better indicators of health risk. So, to give you an example of how this is being applied locally, I'm just going to give a quick example of a source tracking study we did at Little Venice, which is an area here in the Florida Keys that's between Key Colony Beach and Marathon. This is an area that had originally been identified as one of the hot spots of microbial contamination in the Keys. The Keys have a history of septic fields and old cesspits and a couple of local sewers, and it's slowly being sewered along. So it's a real mix of all kinds of sanitary infrastructure. This Little Venice area had been an area of pre-1970s homes, fairly high density along residential canals that was mostly on individual house septic fields and even cesspits. And there was a program put in place to slowly sewer this area of the Keys and to try to improve these known water quality problems. So the Monroe County wastewater plan was to sewer this region, and Little Venice was one of the first areas to go into this program. And this was done in 2004. The problem was, using traditional methods, they didn't see a whole lot of changes in the total enterococci or the total fecal coliforms when tested by the culture method. Part of the problem was they found that some of these organisms were regrowing in the sediments and the mud at the bottom of the canals, on epiphyton and in other areas, and coming from runoff. So there was a disconnect. The current indicators by culture methods weren't really reflecting human fecal contamination. So we came in to do a source tracking study to see if we could find human source fecal markers and to give a better idea if there were changes in the water quality after sewering. So we're looking at the polymerase chain reaction, targeting enterococci groups that are specific to human fecal marker, also Bacteroides that are specific to human marker, as well as some other animal and host markers. So in this test, the Little Venice area, there are 3 canals that were tested that were remediated. These were canals where the homes had been put on a local sewer system. This was a control canal here that had remained on septic, that was not on sewer. Another interesting feature of this 100th Street Canal is there's also a culvert at this end that's a stormwater discharge culvert that takes this whole Marathon Airport area and drains it into this 100th Street Canal. So this canal is still receiving input from septic discharge. These canals are all sewered, but this canal, in addition to being sewered, still gets stormwater discharge. So in this study, we found that all the sewer canals had significantly lower levels of enterococci by the qPCR method than did the canal that was still on septic. So it appeared that there was a decrease in overall human-associated enterococci with the sewered canals, although there was a greater range of variation in the 100th Street Canal, which, as you may recall, was the one that also gets stormwater discharge. In addition, the human-associated fecal markers significantly decreased in the canals that had received sewers. So again, the enterococci levels were higher in the septic canal, and they were also higher with the human source fecal marker in the septic canal, and had decreased in the sewered canals. There was seasonal fluctuation and variation that matched the wet-dry seasons. So in conclusion, there was overall decrease in the sewered canals as compared to the septic canals. Levels of enterococci were also lower in the sewered canals, but had more spikes in the canal that received stormwater discharge. And the other indicators didn't really show a whole lot of difference. So this suggests that the sewer did have a significant impact in improving the water quality of the region. However, from the 100th Street Canal, it's clear that it's not just a septic effect, but you also have to consider your stormwater discharge as another source that's impacting your water quality. Now, what does this have to do with climate change? Well, if we think about the patterns of climate change that we're going to see and sea level rise that we're going to see, the aging sanitary structure we have in the Florida Keys and throughout much of coastal Florida, along the intercoastal waterway, Lake Worth, and a variety of other areas, is going to be very sensitive to very small changes in sea level rise. As you get a small change in sea level rise, it's going to impact how well the area can be drained. It's also going to impact how that septic field is going to be able to function. So the sewering is much more resilient than the legacy septic and cesspit fields. And this study suggests that you get significant improvement in nearshore water quality with the sewering that provides greater resiliency to the community given our expected changes in sea level rise and climate change. So we see a lot of stressors on our local water quality and on our reefs and their impacts on our public health. You know, overfishing, global warming, water pollution, all of this. And what can we do about it? Well, the NOAA mission is one of the missions is to improve our stewardship of our coastal resources and encourage others to get involved, especially in the light of the changes we're expecting to see from climate change, and do what we can to limit the various multiple stressors on our marine environment that are also impacting us. Any questions?